Hey everyone, welcome back. Sorry for the delay. I got my new tool. Let's see if it's broken or not. Hopefully not. All right, so this is where we left off with the last video. So you'll notice I was mentioning earlier how the platelets are so much smaller than the red blood cells. And you guys will learn when you do the reading what these are composed of. And we're going to see when we get into clotting, excuse me, and coagulation, why they're important. These guys, oh no, shenanigans. Oh goodness. I already said this. All right, so these guys from the neutrophil over to the basophil are what are called granulocytes. And my hand is much nicer. My handwriting is much nicer on a board. These guys are called agranulocytes. So what these do, if you see these little dots everywhere, they will release granules. These guys do it for parasitic worms, right? And allergies. Oh, wait, I'm sorry. Parasitic worms and allergies. Oh, no, the thing is doing it again. This is brand new. These guys show up for bacteria. These guys are viruses. Sorry, so we'll put viruses here. Eosinophils are parasitic worms, put a W there, and allergies. And the neutrophils are bacteria. So when you take micro, when we get to the immune system, you'll already have this down, hopefully. Lymphocytes and monocytes, these are, if you're looking under a microscope, you can't differentiate if these are T lymphocytes or B lymphocytes. We'll talk about it when we get to immunity. But the monocytes themselves, these guys will actually grow up and become macrophages, which I'll abbreviate MAC for right now. And these are antigen presenting cells that we will talk about again when we get to immunity. And you'll see them again in not just immunity, but you'll see them in micro when you guys take micro. So it's best to just go ahead and have these under your belt, have them where you know what you're talking about, and you're able to recognize them. So if you look at this, there is an acronym your book mentions that says to remember this. It's never let monkeys, oh my gosh, this is a brand new tool, eat bananas. So neutrophils, lymphocytes, monocytes, eosinophils, basophils. That's fine if you want to do that. But another one that I like my students to know is Ben. I'm going to murder someone. Probably the person, okay, I'm not going to murder anybody, but the person who designed this tool is on my poop box or in my poop list. Okay, so Ben, which is basophils, eosinophils, and neutrophils. And this is from darkest to lightest. So these stain darkest. These are a medium stain, and these are a lightest stain. The coolest thing about the neutrophils is they're the first to arrive. So when, say you've got other things set them off than bacteria, but when you're working on, or when you have an infection, these guys are the first ones to arrive, and then they're followed quickly by the lymphocytes. So, but these are generally what you see if you have acne or you have a pimple. So they're the white part that makes up the pus. So red blood cells, you can see here, they look like little donuts, and we'll see better pictures of them, but they don't have a nucleus. So when they become mature, they spit out the nucleus and become anucleated. And that's also when the hemoglobin proteins form. So keeping that in mind, erythrocytes, again, the red blood cells. So if you see erythro or erythrocytes, the red blood cells, they do have 4 units of hemoglobin. And the hemoglobin itself carries, excuse me, I'm so sorry, guys. Hemoglobin itself will carry, well, that's, yeah, sorry, guys. I'll get to the picture I'll finish this sentence for you guys. Okay, so this is when it's highly oxygenated, it shows a bright red. When it's less oxygenated, it shows up a darker red. But the important thing about red blood cells is they're bringing in O2, okay, to tissues and organ systems that need it, and they're removing CO2. I cannot believe I wasted my money on a new tool. Yay, me. Okay, so they bring in CO2, or I'm sorry, they bring in O2 and they remove CO2 from cells and tissues. So that as the cells respire and everything like that, and they use up the oxygen, this oxygen can be replaced by the red blood cells, okay? And the CO2 itself can be removed since it's a waste product, and that can mess with your carbonic acids and carbon anhydrase enzymes. All right, so again, you don't need to memorize these numbers. Just recognize that males typically have larger numbers of red blood cells because typically they're larger than women. Okay. All right, so the hemocrit is a pretty nice table in your book on table 19.1, right? 19, oh my organs. All right, and it talks about how these numbers are calculated. So just be aware that if you, and it's on it's table 19.1 on page 665 in the textbook. And it talks about how you can calculate these. So, let's see. All right, so these are definitely highly specialized cells. They don't, like, again, I mentioned they lose their nucleus. They also don't have mitochondria. So when I said they drop their nucleus, oh my gosh, it's supposed to be an N. They also drop their mitochondria. And so they're basically forced to use anaerobic respiration. Otherwise, if they had mitochondria, they would use up all the O2 before they even got to deliver it. So this is why they drop both the mitochondria. The nucleus, they drop because when we get to the structures of these guys here, they're so busy carrying the heme and alpha beta genes that they cannot, they don't have room for the nucleus. So that's why they drop these organelles, okay? Let's see. So if you were in lab with me, you would see these, and we would look and see, like, these little tiny dots are actually platelets. Okay, the big ones that look like donuts with the little white hole, or this one, it doesn't even have a little white hole. The big ones are red blood cells. I don't see any white blood cells on this slide. Nope, I don't see any. But again, these little ones wouldn't be counted when you're looking at blood because there are red blood cell counts, or packed volume counts. There's mean corpuscular volume and another one, but they wouldn't be, platelets aren't counted in that. It would be solely the red blood cells. So here's what they look like if you do not have sickle. Oh my gosh, where is? If you don't have sickle cell disease, or sickle cell anemia is what I call it. This is what they look like. All right, so again, if you take a cross section of this is their entire function, and they're pretty small, but there's like 75 billion of them in each human being, which is pretty insane. These guys make up, but they've got a lot of work to do. And remember, they're a connective tissue with the matrix they provide. So they have a very large surface area to volume ratio, so that they can very quickly absorb O2 when they're in the lungs, and then quickly release it to the tissues. The relu, relu plural, is how they actually form little stacks, and I'll show you a picture. But they stack up like plates, like here, this little stack, so they can get through these little tiny capillaries. They look ginormous here, but these are section capillaries, and they are magnified by a factor of 1,430. So they're just showing you how they get through. Now, that doesn't work as well when you have sickle cell anemia, because instead of the cells being this rounded biconcave shape, they're actually a sickle shape. So, and then when they stack up, oh my gosh, I cannot believe it. When these guys stack up, they get stuck in the capillaries, and this can lead to extreme pain. And I'll talk briefly about the treatments for sickle cell later. Okay, so when they're not sickled, they can bend and flex through capillaries and get through, which we've already talked about how they stack up. But these are sectioned out capillaries. You can see how they stack up and get through. And just imagine if all of them were this shape, right? They would just get stuck together in these capillaries, and they're not as. These guys are flexible. They're biconcave discs, and we're talking about normal red blood cells or typical red blood cells. When they are sickled, they are not able to flex. Okay, and they are far more rigid. All right, so here's where I was talking about the anucleation, or they spit out their nuclei. They also don't have mitochondria, right? And they also lack ribosomes. So they lack the majority of organelles that typical cells have. And if you're watching some kind of crime show and they say, oh, we found blood on the scene and we're going to get a DNA sample, the DNA sample doesn't come from these guys. It comes from white blood cells. That's where the DNA sample comes from. So red blood cells typically live about 120 days, which is kind of short when you're looking at lifespans for cells in our body. But it's because they get used so frequently. And then we'll see later in the chapter how you guys, or how they're broken apart once they are past being used. Also, I said mitochondria, I said nucleus. Oh, and then once they're mature, the other thing for them to be mature is they are going to start to form the hemoglobin itself, which we'll get into in the next picture. All right, so I already mentioned that DNA is collected from white blood cells, not red blood cells. And then, even if you find somebody's broken hair at a crime scene, that's just keratin itself. You get the DNA from the hair follicles. Okay, so hemoglobin, you can abbreviate it HB or HGB. It's up to you. The adult male has typically more hemoglobin grams per deciliter than the adult female. That's nothing uncommon, right? So because they have larger ones. So we were talking about 2 units, the alpha and the beta. The 2 units for alpha and beta are going to bind. Where's my little picture? Yeah. So if you have 2 alphas and 2 betas, that gives you 4 chains total, and they're going to bind the CO2, okay, and get rid of it. So this is going to bind the CO2. The hemoglobin, the hemocrit, which is shown here, the heme group right there. I should say heme group, not hemocrit. Sorry, the heme group right here. There are 4 of them, and these actually bind O2. So that's an O and a 2, oxygen, blah. All right, so they're going to bring in oxygen. That's what the heme does. Please know this. And while the alphas and the betas are going to remove CO2. I cannot believe I wasted my money on this tool. Okay. All right, iron interacts with oxygen. When that happens, you have oxyhemoglobin. So this would be abbreviated HbO2. Right. And then when it's dissociated so this would be the bright red blood cells. The darker red blood cells will be the deoxyhemoglobin after they've dropped off their O2 with the tissues. So the only other thing I was going to say is in sickle cell anemia, normally, and I'm not going to beat you guys up with how many amino acids are in each chain, but normally, so if you were to just draw this with an alpha here, and an alpha here, and a beta here, and a beta here, these chains usually have a charge. So with it is a point mutation in the genetics. These, instead of having charges and repelling each other, is negative. So they will repel because opposites repel. No, opposites attract. Sorry. Legs repel. They don't repel because the charge is removed in the sickle cell. And then you end up with, instead of a biconcave disc, you end up with a sickled red blood cell. More information you guys ever wanted, probably. All right, so hemoglobin in embryos or a fetus, it actually has an affinity to bind oxygen better than does adult hemoglobin. So one of the treatments for sickle cell anemia that they've come up with is a gene therapy to replace the adult hemoglobin gene. We'll just call it adult HG they'll replace it with a fetal hemoglobin gene. And this has actually been successful in at least one trial. I think it took them on the order of 6 or 8 years to get the gentleman who volunteered for this trial to actually have him express entirely fetal hemoglobin and overcome his sickle cell disease. But it definitely worked for him. He reports way less pain, and this is all pre-pandemic, but they were working on bringing this gene therapy out for wider scale use for sickle cell anemia. Okay, so the function for hemoglobin, hemoglobin function. Every red blood cell has about, you don't need to memorize all these numbers, but millions of hemoglobin molecules. And this one just shows here, excuse me. So just imagine like this is just one little part. So anyways, okay, they do bind oxygen. Nope, that's not it. And they'll carry over 1 billion molecules of O2 into the tissues and get them where they release CO2, and then they'll bind CO2 again. Once it's bound the CO2, we're going to use the carboamino hemoglobin word. It's not very fun, but just remember CO2 is bound. It's got a carboamino in there. Okay, so when the blood gets to the lungs, oxygen is hopefully really high with its billions of hemoglobins. It's going to release the CO2 when you breathe out and take in the O2 when you breathe in. I hope everybody gets that part. All right, so when we get into anemias, there's a whole shenanigans. Is there no eraser anymore? Let's try this way. Sorry, guys. All right. What a pain in the neck. What a waste of my money. All right, okay. So when okay, so there's a bunch of different types of anemia. There's a table somewhere. Because when you guys get to the case studies, you're going to want them. Where is the anemia one? Oh, is it a different book? No, I thought it was in here. Nope. Sorry, guys. I thought it was right here. Oh, it's clotting factors. Okay, it was a different book. But there's a bunch of different types of anemia. Basically, if you hear something anemia, it means that the hematocrit or hemoglobin is reduced. Okay, it could be sickle cell anemia, right? It could be induced when women have their menses and they are losing the lining of their uterus and their iron drops. But anyways, anemias definitely interfere with oxygen. And when we have the red blood cells have a turnover, you get these guys. Where did I go? Sorry, my phone is bothering me. All right, so when we have the turnover of the red blood cells, you get one of them circulating, and then 1% of them get replaced every single day. Right? Remember, they have a short lifespan of 120 days. So, and then every second, if everything was going well and you're a healthy young adult, you're not geriatric, which is elderly, you've got about 3 million new red blood cells coming into this bloodstream every single second. Okay, so this is a cool little figure. I like this guy. So we're talking about erythropoiesis, sorry. And then you're going to see later, we're going to be talking about erythropoietin, right? EPO. It helps with the formation of red blood cells specifically. Oh my gosh, I'm not from kindergarten, I swear. Okay, but erythropoiesis, these guys, is how we're going to start with this day one. There's a far more complicated figure in your book. If you guys are familiar with uh, the anucleation ejection of the nucleus and the formation of the hemoglobin proteins here, that's good for this figure. When we get to the next figure, well, not the next one, but when we get to the figure that shows both red blood cells and white blood cells, I'm going to help you guys walk through you guys what you really do want to know. We're going to get into the point with both of these where they differentiate into stem cells. I'm not sure why this guy stops working every once in a while. Where they differentiate, from red blood, from stem cells. And then they go through several processes to get to produce your red blood cells. So here's a mature guy. He has no nucleus. He got ejected. He has no mitochondria, but he does have hemoglobin, right? This in adults, which is where you guys definitely want to know it, it's going to happen in the red bone marrow, which is primarily in your hip bones and your femurs. I'm going to stop here because I feel like I've been babbling forever.